to this presentation, which is entitled Prognostic Impact of CHIP in the General Population. My name is Joop JANSEN and I work at the Radboud University in the Netherlands, and unfortunately we cannot meet live in Nantes, but I hope that this presentation will be useful for this conference. As a background to the work I will present, first I will show a few slides on a relatively new phenomenon that emerged in 2014. Around that time, several papers were published that showed that the age-related emergence of expanded hematopoietic clones carrying acquired leukemia-associated mutations can very often be observed in healthy individuals. These clones are usually expanded to a size of 2 to 5% of all hematopoietic cells, which is already quite large, but often they do not create any health problem. Still, the presence of these clones correlates with an increased risk of cardiovascular disease or the development of hemato-oncological malignancies. In the last few years, this process has been studied in detail in hematological cells. We know that random mutations occur in our genome, for instance by toxic agents, UV irradiation, and mistakes made during the copying of the DNA during cell division. For instance, each cell division induces about 2 to 10 mutations in our genome. As a result, in adults, every cell contains about 1,000 to 1,500 acquired random mutations, and since approximately 2% of our genome is coding for proteins, which is called the exome, this implies that about 10 to 20 mutations will be present in the exome in an adult person. Most of these mutations are biologically inactive and not harmful. Still, they are passed on to all daughter cells, and these mutations are called passenger mutations. When, however, a mutation occurs in a gene that regulates the balance between proliferation and differentiation, the cell may acquire a growth advantage, leading to the expansion of a clone that harbors the mutation. These mutations are called driver mutations. Clonal expansion does, however, not necessarily lead to clinical symptoms. The acquisition of additional mutations may, however, eventually lead to malignant transformation. As a result, non-malignant but already vastly expanded clones carrying 1 or 2 driver mutations can frequently be found in healthy individuals in many different tissues, and this strongly increases with age. When this occurs in hematopoiesis, we call this clonal hematopoiesis of indeterminate potential, or CHIP, and it's called indeterminate since in most cases this does not develop into a disease, but in other cases it does. Although in many cases CHIP is not harmful, nonetheless individuals with CHIP have a 10-fold higher chance of developing a hemato-oncological disease and about 3 to 4-fold higher chance of developing cardiovascular problems. This latter is probably due to pro-inflammatory effects of the mutated cells initiating local coagulation defects. Now, as anemia is frequently observed in elderly persons and increases with age, we ask ourselves to what extent is anemia in the elderly in fact undiagnosed MDS, or is it a pre-stage of MDS, and what is the possible role of CHIP? For this we made use of the Lifelines cohort, which is a population-based cohort in which 167,000 persons participated and from whom DNA material, blood values and health parameters are available. From this cohort we selected all persons who were 6 years or older and who were anemic by the definition of the WHO, having a hemoglobin level of less than 30 grams per deciliter for men and less than 12 grams per deciliter for women. This yielded 676 anemic individuals, and from these we were able to perform next-generation sequencing for myeloid malignancy-related genes in 635 cases. We also selected a control group of non-anemic persons who were matched for age, sex, BMI, and the number of medications, and for this group we were able to generate next-generation sequencing data for 663 cases. Finally, we could collect DNA and data from these persons at a median follow-up time of 4.5 years, as indicated in the red box. We performed error-corrected next-generation sequencing for 27 myeloid driver genes, allowing us to detect somatic mutations with a variant allele frequency from 1% and larger, which corresponds to clone size of 2% and larger, and that is a little bit more sensitive than in some of the previous large cohort studies. This graph shows the presence of chip clones with a size of 1% or larger in the non-anemic control group. And you can appreciate that at the age of 60, already 30% of individuals carry chip clones, and this increases to about 65% above the age of 85. Here you can see that in anemic persons, here depicted in red, chip clones can be more frequently found than in non-anemic matched controls in blue, but the difference is maybe a little bit less than we would have anticipated before we started this study. This does already indicate that anemia in the elderly is not entirely explained by the presence of mutated clones, and hence that not all anemic cases should be considered as pre-stage MDS. This figure shows the different mutations in chip clones as they were found in the anemic cases on top in red and the controls at the bottom in blue. At the y-axis you can see the different genes that may be mutated, and at the x-axis are all the individual cases. The color gray indicates that no mutation was found in that gene, and red or blue indicates the presence of a mutation, and the dark red and dark blue indicate biallelic mutations as we see them often in DNMT3A and TET2. When you look at the more frequently mutated genes, being the DNMT3A, TET2 and ASXL1 genes, you can see that between anemic cases and controls there does not seem to be a very big difference. In contrast, when you look at the genes that are less frequently mutated, these appear to be less often mutated in the controls at the bottom compared to the anemic cases in the upper part of the graph. When looking more in detail to particular genes, you can see that mutations in, for instance, DNMT3A and TET2 are equally distributed between anemic and control cases. However, TP53 and SF3B1 mutations were enriched in the anemic cases. In addition, more rarely mutated genes like NRAS and KRAS were overrepresented in anemic cases, but due to the low numbers, statistical significance was only reached when these rare genes were grouped together, here indicated as other genes. Finally, JAK2 mutations appear to be overrepresented in the non-anemic cases, but due to the low numbers, also here statistical significance was not reached. When looking at clone sizes, you can see that in general, the variant allele frequency was around 3%, which would translate into 6% of all the cells being mutated. However, you can also appreciate that there were quite a number of cases where clone sizes were above 10%, up to 50%, which would mean that there were 20 to 100% of the cells being part of the chip clone. What you can also see is that there was not a very big difference between anemic and control cases. In the anemic cases, the median VAF was somewhat higher than in the non-anemic controls, with a median VAF of 3.35% in anemic cases versus 2.8% in the non-anemic controls. This trend was not statistically significant, but it seems to indicate that overall in anemia, clone sizes are a bit larger, in the order of 1%, which is certainly much less than we would have expected. In most cases with CHIP, only one gene was found to be mutated, but also cases with 2, 3 or 4 mutated genes were found. As you can see, the number of cases with one or multiple mutations did not differ between anemic and non-anemic cases. For many cases, follow-up samples were available, allowing us to follow clonal growth over time. The median overall VAF increased by about 0.5% in 44 months, but when looking at individual cases, growing clones were seen above the gray line here indicated, but interestingly also many cases were observed showing declining clone sizes. Overall, CHIP mutations therefore provide a growth advantage, but in many individual cases also important regression of CHIP clones may be observed. The factors that underlie this phenomenon are as yet unknown, but obviously we are very much interested to study this further. In this graph you can see the growth speed of clones according to the mutations that they harbor. The size of the circles indicates the number of cases with a specific mutation, with the most frequent mutations being DNMT3A in light green and TET2 in purple. The x-axis depicts the clonal growth expressed in variant allele frequency, and the y-axis indicates the p-value, indicating whether the growth is significantly different from zero or not. What is quite striking is the fact that the most frequently found CHIP mutation, which is DNMT3A, these clones hardly show any growth over a period of 44 months. And apparently, cells that acquire a DNMT3A mutation, they do grow out to occupy 1 to 5% of all hematopoietic cells, but apparently they do not expand much further. This is quite counterintuitive, and the mechanisms that initially allow clonal outgrowth but subsequently control the clone size remains unknown, and also this is an area in which we will certainly further investigate. As you can see, clones that contain a JAK2 mutation are the fastest growers, followed by clones that harbor SRSF2 or U2AF mutations. Next, we looked into the mutational spectrum in the commonly used subtypes of anemia, being nutrient-deficient anemia, anemia of chronic disease, and unexplained anemia. We noted a higher incidence of DNMT3A mutations in anemia of chronic disease, a higher incidence of TP53 mutations in unexplained anemia, and a overrepresentation of rare mutations, here called other mutations, in anemia of chronic disease as well as in unexplained anemia. Overall, however, the mutational spectra in the three different types of anemia was quite comparable. Finally, we looked at overall survival. The best overall survival was seen in non-anemic controls without CHIP, depicted here in light blue. Slightly worse overall survival was seen in the non-anemic controls who harbored CHIP clones, depicted by the dark blue line. For anemic cases without CHIP, depicted by the orange line, overall survival was better than for individuals with anemia and CHIP, as shown by the red line. And these data indicate that CHIP confers a worse overall survival both in anemic cases as well as in the non-anemic controls. Overall, however, the differences in survival were not very large. More important differences in overall survival were, however, seen when clone size was taken into account. Individuals without clone hematopoiesis, depicted in the gray line, had a similar overall survival compared to individuals with CHIP clones smaller than 5%, depicted in the orange line. In contrast, CHIP clones larger than 5% correlated with a clearly inferior overall survival, as depicted by the red line. When looking at the number of genes that were mutated within one individual, no difference was observed in overall survival if 0, 1 or 2 mutations were present. However, when 3 or more mutations were found, here depicted in red, this correlated with poor overall survival. Finally, we looked at the probability of developing a hematological malignancy in the presence of anemia with and without CHIP. On the y-axis, you can see cumulative mortality, and in red, death from hematological malignancies is indicated. In the upper part, you can see that without clonal hematopoiesis, anemic persons show a higher mortality rate than non-anemic persons, represented by a higher gray area under the curve, but no deaths were seen due to hematological malignancies. In the presence of clonal hematopoiesis, also anemic persons did worse, and in addition, the presence of CHIP correlated with several cases who died from a hematological malignancy. In other words, in the absence of CHIP, the chance of dying from a hematological malignancy are very small, but in the presence of CHIP, this may occur, and the risk of developing a hematological malignancy is higher when also anemia is present. With that, I would like to end, and that brings me to the conclusions. Community-dwelling individuals who are anemic have a higher incidence of CHIP, but the difference with non-anemic controls is not very large. Second, the mutational spectrum of anemic individuals is enriched for TP53 and SF3B1 mutations. Third, clone sizes in anemic persons are slightly higher compared to non-anemic controls. Overall, CHIP clones expand in time, but surprisingly, many individual cases may also show strongly regressing clones. The underlying mechanisms remain unknown. Paradoxically, clones with the most frequently found chip mutation DNMT3A show the lowest growth rate. Furthermore, smaller chip clones do not confer an important effect, but clones with a VAF of 5% or more correlate with a clearly worse overall survival. The presence of chip may predict a higher chance of developing a hemato-oncological disease, and this effect is stronger when also anemia is present. And finally, in contrast what might have been expected, chip can only partially explain unexplained anemia. Thank you very much for your attention, and in this slide I would like to acknowledge the people who have contributed to this work. Thank you very much.